I'm excited to welcome you all to this lecture on humoral and cell-mediated immunity. Today, we're going to explore two critical components of the adaptive immune system, which together provide a highly specific and powerful defense against infections and abnormal cells. Before we start, I want to emphasize how fascinating and vital the immune system is, not only as a protector of our health, but also as a model of biological complexity and precision. Our immune system isn't just a random collection of cells and molecules. It's an intricately coordinated network that can distinguish friend from foe, remember past invaders, and tailor its response accordingly. Today, we'll focus on the adaptive immune system, specifically its two main arms: humoral immunity, which involves B cells producing antibodies to neutralize pathogens outside cells, and cell-mediated immunity, where T cells seek out and destroy infected or abnormal cells. Understanding these two arms is essential not only for appreciating basic biology, but also for applications in medicine like vaccine development, immunotherapy for cancer, and managing autoimmune diseases. So, over the next 30 minutes, we'll take a detailed journey through the cells, molecules, and mechanisms that make these immune responses so effective. Feel free to ask questions anytime, and I encourage you to engage actively because this is a rich and complex topic that rewards thoughtful discussion. Let's begin our exploration. Before diving into the details, let's look at what we aim to cover today. This overview will help you keep track of the flow and the main takeaways. First, we'll start with a brief recap of the immune system, focusing on how adaptive immunity fits within the broader immune defense strategy. We'll understand the difference between innate and adaptive immunity and why adaptive immunity is so crucial for specific, long-lasting protection. Then, we'll turn our attention to humoral immunity. We'll discuss how B cells recognize specific antigens, the process of their activation, differentiation into plasma and memory cells, and the critical role antibodies play. We'll explore the structure of antibodies, their classes, and their mechanisms of action, how they neutralize pathogens, tag them for destruction, and activate the complement system. After that, we'll shift focus to cell-mediated immunity. We'll learn about T cell development and activation, the various types of T cells, helper, cytotoxic, and regulatory, and their functions. We'll discuss how T cells recognize infected or abnormal cells, coordinate immune responses, and maintain immune balance. Next, we'll compare humoral and cell-mediated immunity, highlighting their differences, similarities, and how they work synergistically to provide robust defense. Finally, we'll discuss clinical relevance, how knowledge of these immune responses informs vaccine development, explains immune deficiencies such as HIV, and helps us understand autoimmune diseases and immunotherapies. By the end of this lecture, you should have a thorough understanding of both arms of adaptive immunity and their importance in health and disease. To appreciate humoral and cell-mediated immunity fully, we need to start with a quick review of the immune system's overall organization. Our immune system has two fundamental components: innate and adaptive immunity. Innate immunity is the body's first line of defense. It includes physical barriers like the skin and mucous membranes, chemical barriers such as stomach acid and enzymes, and cellular defenses, including phagocytic cells like macrophages and neutrophils, as well as natural killer cells. The innate system responds quickly but nonspecifically. It can't distinguish one pathogen from another and doesn't develop memory. In contrast, adaptive immunity is slower to activate but highly specific and tailored to particular pathogens. It develops over days to weeks after exposure and crucially generates immunological memory, meaning the immune system remembers pathogens it has encountered before, resulting in faster and more potent responses upon re-exposure. This feature is the foundation for effective vaccination. Adaptive immunity primarily involves two types of lymphocytes, B cells and T cells. B cells are responsible for producing antibodies in the humoral immune response, targeting pathogens found outside cells, like bacteria and toxins. T cells handle cell-mediated immunity, targeting infected or abnormal cells, especially those harboring viruses or cancerous changes. An important concept here is antigen recognition. Antigens are foreign molecules, usually proteins or polysaccharides, present on pathogens or abnormal cells. Lymphocytes have specialized receptors that recognize specific antigens, allowing the immune system to distinguish self from non-self and respond precisely. Understanding these foundational concepts prepares us to explore the detailed workings of humoral and cell-mediated immunity, which we will do in the next sections. Adaptive immunity divides into two main branches, humoral and cell-mediated immunity. Both have distinct but complementary roles. Humoral immunity involves B lymphocytes that circulate in blood and lymph, producing antibodies, specialized proteins that bind to antigens on extracellular pathogens or toxins. These antibodies neutralize pathogens, preventing them from invading cells, and flag them for destruction by other immune cells. Cell-mediated immunity is driven by T lymphocytes, which do not produce antibodies but play critical roles in identifying and eliminating cells harboring intracellular pathogens like viruses or certain bacteria. T cells also regulate immune responses and maintain tolerance to avoid attacking the body's own tissues. There are different subsets of T cells, including helper T cells, CD4+ coordinate immune responses by secreting cytokines. Cytotoxic T cells, CD8+, directly kill infected or abnormal cells. Regulatory T cells suppress immune reactions to maintain balance and prevent autoimmunity. Think of humoral immunity as the immune system's patrol on the body's surfaces and fluids, while cell-mediated immunity is the tactical force that eliminates hidden threats inside cells. Both arms are essential, and their coordination is vital for effective immunity. Let's focus now on humoral immunity, the defense mechanism against pathogens present in extracellular spaces like blood, lymph, and tissue fluids. The central players are B lymphocytes. Each B cell expresses a unique B cell receptor, BCR, on its surface, which is essentially a membrane-bound antibody specific to a particular antigen. When a B cell encounters its matching antigen, it internalizes it and presents fragments on its surface via MHC class II molecules. This presentation allows interaction with helper T cells, which provide additional activation signals, ensuring the immune response is appropriate and controlled. Once fully activated, B cells proliferate and differentiate into plasma cells, antibody-producing factories that secrete large amounts of soluble antibodies into the bloodstream. Memory B cells, long-lived cells that persist in the body, ready to mount a rapid response if the same antigen appears again. Antibodies produced by plasma cells circulate freely and bind to pathogens, neutralizing them or marking them for destruction by phagocytes. Humoral immunity is particularly effective against extracellular bacteria, viruses before they enter cells, and soluble toxins. It is also the basis of protection provided by many vaccines. Let's dig deeper into the process of B cell activation and differentiation, which is a multi-step and highly regulated process ensuring precision. Initially, a B cell's receptor binds a specific antigen. This binding alone can activate B cells in some cases, especially for T-independent antigens like polysaccharides, but most often, full activation requires help from helper T cells. This is called T-dependent activation. Helper T cells recognize the antigen presented by B cells via MHC class II molecules and, in turn, provide co-stimulatory signals through surface molecules and cytokines. This two-step activation prevents inappropriate antibody production. Following activation, B cells undergo clonal expansion, rapid proliferation producing many identical cells. Some differentiate into plasma cells, which migrate to bone marrow or lymph nodes and secrete vast amounts of antibodies. Others become memory B cells, which circulate long-term and ensure a faster, stronger response if the same antigen is encountered again. This mechanism underlies immunological memory and vaccine effectiveness. Additionally, during this process, B cells undergo class switching, changing the antibody isotype they produce, e.g., from IgM to IgG, to tailor the immune response to different pathogens and tissues. Finally, B cells also undergo affinity maturation, where mutations improve the binding affinity of antibodies for their antigen, enhancing the immune response's quality. Antibodies, or immunoglobulins, are Y-shaped proteins composed of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains. The tips of the Y-shaped molecule contain the variable regions, which form the antigen binding sites. These regions are unique for each antibody and determine specificity toward a particular antigen. The stem of the Y is the constant region, which defines the antibody's class and mediates effector functions. There are 5 major antibody classes. IgG, the most abundant antibody in blood and tissues, crucial for long-term immunity and opsonization. IgA, found in mucosal secretions like saliva, tears, and breast milk, protecting mucosal surfaces. IgM, the first antibody produced in an initial immune response, exists as a pentamer, making it highly effective at agglutinating pathogens. IgA, involved in allergic responses and defense against parasitic infections. IgD, primarily found on naive B cells, its function is less well understood. Antibodies protect by neutralization, blocking pathogens or toxins from binding to host cells. Opsonization, coating pathogens to promote phagocytosis. Complement activation, triggering a cascade that leads to pathogen lysis and inflammation. Understanding antibody structure and function helps us grasp how vaccines and antibody-based therapies work. Humoral immunity employs several mechanisms to eliminate extracellular pathogens. Neutralization. Antibodies bind to viral particles or bacterial toxins, preventing their interaction with host cells. For example, neutralizing antibodies block viral entry into cells, effectively halting infection. Opsonization. Antibodies coat microbes, marking them for ingestion and destruction by phagocytes, such as macrophages and neutrophils. Phagocytes have Fc receptors that recognize the antibody's constant region, enhancing pathogen uptake. Complement activation. Antibodies bound to pathogens initiate the classical complement pathway. Complement proteins form membrane attack complexes that puncture bacterial membranes, causing lysis. Complement fragments also attract inflammatory cells, amplifying the immune response. Agglutination and precipitation. Multivalent antibodies cross-link pathogens or soluble antigens, forming aggregates that are easier for phagocytes to clear. These mechanisms work in concert to prevent pathogens from spreading and to promote their clearance from the body. Now, let's shift our attention to cell-mediated immunity, which combats pathogens that hide inside host cells, viruses, some bacteria, and cancer cells. The main actors here are T lymphocytes. Unlike B cells, T cells don't produce antibodies. Instead, they identify infected or abnormal cells by recognizing antigen fragments presented on the surface via MHC molecules. There are 3 major T cell subsets. Helper T cells, CD for plus, they orchestrate immune responses by secreting cytokines that activate macrophages, B cells, and cytotoxic T cells. They're the generals of the immune system. Cytotoxic T cells, CD8+, these cells directly kill infected or abnormal cells by inducing apoptosis. Regulatory T cells, they suppress immune responses to maintain self-tolerance and prevent autoimmune damage. Cell-mediated immunity is essential because many pathogens evade antibodies by hiding inside cells. Only T cells can recognize these hidden threats and eliminate them. T cells develop from hematopoietic stem cells in the bone marrow but mature in the thymus, where they undergo rigorous selection processes to ensure they can recognize foreign antigens but not attack self-tissues. During development, T cells randomly rearrange their T cell receptor, TCR, genes to generate diverse antigen specificity. Positive selection in the thymus ensures T cells can recognize self-MHC molecules, while negative selection removes those strongly reactive to self-antigens, preventing autoimmunity. Once mature, naive T cells circulate through lymphoid organs, searching for their specific antigen. T cell activation requires 2 signals. Antigen recognition. The TCR binds a specific peptide antigen presented by MHC molecules on antigen-presenting cells, APCs, such as dendritic cells. Co-stimulation. Additional interactions between surface molecules on T cells and APCs provide necessary activation signals. Helper T cells recognize antigens on MHC2 molecules. Cytotoxic T cells recognize peptides on MHCI molecules. Following activation, T cells proliferate and differentiate into effector and memory cells, ready to perform their functions. Helper T cells are the immune system's conductors, coordinating the activity of many other immune cells. Upon activation, they secrete various cytokines, chemical messengers, that influence the behavior of B cells, cytotoxic T cells, macrophages, and other immune cells. Helper T cells differentiate into subsets. Th1 cells promote cell-mediated immunity by activating macrophages and cytotoxic T cells, crucial against intracellular pathogens like viruses and some bacteria. Th2 cells support humoral immunity by helping B cells produce antibodies, especially IgA, involved in responses to parasites and allergens. Th17 cells involved in inflammatory responses, helping fight extracellular bacteria and fungi. By producing the right cytokines, helper T cells tailor the immune response to the nature of the invading pathogen, ensuring an effective defense without excessive damage. Cytotoxic T cells are the immune system's assassins. They patrol the body, inspecting cells for signs of infection or abnormality. They recognize antigen fragments presented by MHCI molecules, which are expressed on almost all nucleated cells. When a cytotoxic T cell identifies an infected or cancerous cell, it releases perforin, which forms pores in the target cell membrane, and granzymes, which enter through these pores and induce apoptosis, programmed cell death. This targeted killing eliminates the pathogen's cellular niche without triggering widespread inflammation or damaging neighboring healthy cells. Cytotoxic T cells are also involved in transplant rejection and responses to tumors, making them a key focus of immunotherapy research. Regulatory T cells, or TREGs, are the immune system's peacekeepers. Their main role is to suppress excessive or inappropriate immune responses that could damage the body's own tissues. TREGs help maintain immune tolerance, the ability of the immune system to avoid attacking self-antigens. They secrete immunosuppressive cytokines like IL-10 and TGF-beta and can directly inhibit the activation and proliferation of other T cells. Without regulatory T cells, autoimmune diseases, where the immune system attacks its own body, would be much more common. TREGs also help resolve inflammation after infection and play roles in maintaining tolerance to harmless environmental antigens, like those from food or commensal microbes. Now, let's compare humoral and cell-mediated immunity side by side. Humoral immunity is primarily directed against extracellular pathogens in body fluids. It involves B cells and antibodies that neutralize pathogens, promote phagocytosis, and activate complement. Cell-mediated immunity targets intracellular pathogens and abnormal cells. It involves T cells that kill infected cells or regulate immune responses. Humoral immunity acts mainly in body fluids, whereas cell-mediated immunity acts within tissues and cells. While both systems are specific and generate immunological memory, their mechanisms differ. One uses soluble antibodies, the other uses cellular cytotoxicity and cytokine signaling. Importantly, they work together. Helper T cells activate B cells in humoral immunity, and antibodies can assist cytotoxic T cells by marking targets. This synergy ensures a comprehensive immune defense capable of handling a wide range of threats. Understanding humoral and cell-mediated immunity has profound clinical implications. Vaccines leverage these mechanisms. Some vaccines, like the tetanus toxoid, induce strong antibody responses, while others, like BCG for tuberculosis, primarily stimulate T-cell-mediated immunity. Modern vaccines aim to elicit both arms for optimal protection. Immunodeficiencies provide insight into immune function. HIV infects helper T cells, crippling cell-mediated immunity and leading to AIDS, characterized by susceptibility to opportunistic infections. Autoimmune diseases occur when immune regulation fails and the immune system attacks self-tissues. Conditions like lupus, rheumatoid arthritis, and type 1 diabetes involve both humoral and cell-mediated mechanisms. Immunotherapy for cancer, such as checkpoint inhibitors and CAR T cells, harness cell-mediated immunity to recognize and kill tumor cells. By understanding these immune responses, clinicians can better diagnose, treat, and prevent disease. To summarize, adaptive immunity consists of two main branches: Humoral and cell-mediated. Humoral immunity involves B cells producing antibodies that neutralize extracellular pathogens and toxins. This arm is crucial for defense in body fluids and forms the basis for many vaccines. Cell-mediated immunity involves T cells that identify and eliminate infected or abnormal cells. It's essential for clearing intracellular pathogens and cancerous cells. Helper T cells play a central coordinating role, activating both B cells and cytotoxic T cells, while regulatory T cells maintain immune balance. Together, these systems provide a precise, adaptable, and powerful defense that protects us from infection and disease. Understanding these mechanisms is fundamental for advances in medicine, including vaccines, immunotherapies, and treatments for immune disorders. Finally, for those interested in delving deeper, here are some key resources. I encourage you to explore these materials and stay curious, as immunology is a rapidly evolving and fascinating field. Make sure to subscribe for more content. See you.